The big idea in stage 2 of glycolysis is the phosphorylation of ADP to form ATP. And in essence, in order to make this work, conceptually and from a fundamental organic chemistry perspective, what we need to do is get the phosphate group connected to something that is a good leaving group. Good in the thermodynamic sense, something that is okay departing from phosphate with a pair of electrons. In essence, what happens when we phosphorylate ADP is that the terminal phosphate acts as a nucleophile toward phosphorus as an electrophile in this high-energy organophosphate intermediate. And the way we encourage that to occur is by converting whatever the phosphate is attached to into a good leaving group. As long as that leaving group is good, this reaction is going to favor the product side. And that's essentially our goal, right? The synthesis of ATP is the overarching energetic goal of glycolysis. The question really that we should have as bioorganic chemists is, what is the nature of this species more specifically? What is the structure of this leaving group? What kinds of structural factors tell us that it's a good leaving group? And that will become apparent as we work through stage 2. And how do we transform glyceraldehyde 3-phosphate, G3P, which definitely does not meet this condition, right? Since this leaving group would be an alkoxide, that's a very poor leaving group. How do we convert this into something that does contain a good leaving group linked to phosphate? Well, actually, the very first step of stage 2 starts to get at this. This step involves what is essentially a nucleophilic acyl substitution. And in order to illustrate that, what I want to highlight are the things connected to the carbonyl carbon of G3P, a hydrogen, and the product, a phosphate group. Notice that in essence, what has happened here is the substitution of phosphate, which is a good nucleophile. Recall that the oxygens in the phosphate anion are decently good nucleophiles with negative charge. That substitutes for another nucleophile, quote unquote H-, right? Which we don't see on the product side. We don't see it over here because it becomes incorporated into NADH. And we can actually think of NADH as a kind of nucleophile in its own right because it's a biochemical or biological source of H-, which is the nucleophilic hydrogen or hydride anion. On the whole here, viewed from a bird's eye view, what we're doing in this 2.1 step of glycolysis is nucleophilic acyl substitution, where NAD+ is accepting the hydride leaving group. And the whole point of this, again, is to establish a good leaving group connected to a phosphate within a metabolite. And that's exactly what we see on the product side here. But before we talk in more detail about that, let's look a little bit more in detail at G3P dehydrogenase, the enzyme that catalyzes this process. Here's the enzyme's active site, and we can see the usual smorgasbord of intermolecular forces holding the anionic phosphate group in place and interacting with the aldehyde carbonyl group, for example. There are all kinds of intermolecular forces that hold the substrate in place. A naive nucleophilic acyl substitution mechanism would be something like direct addition of phosphate to the carbonyl carbon through one of its oxygens, followed by, in like an ADN step, right? Followed by beta elimination of hydride directly back to NAD+. And while that's not necessarily energetically forbidden, it may not be the lowest energy path. In G3P dehydrogenase, a cysteine residue gets involved. Actually, in this active site picture, the key cysteine has been mutated to an alanine. In the actual wild type or natural enzyme, this is a cysteine residue that is deprotonated to form a good nucleophile. And a key step, the key first step really of this process, is the addition of that cysteine to the carbonyl carbon of glyceraldehyde 3-phosphate. These arrows within the active site picture might be a little bit easier to see over here. We get an addition of that negatively charged cysteine residue to the aldehyde carbonyl carbon. And this actually facilitates elimination because the thing that happens next is elimination, and I'm just going to kind of condense the curved arrows, of H to NAD+. And so rather than phosphate acting as the nucleophile directly, a cysteine residue acts as the nucleophile. Because hydride, hydrogen and a pair of electrons, the pair in the aldehyde CH bond, was transferred to NAD+, this generates NADH, and it also generates a relatively high energy thioester, intermediate. Sulfur has substituted for the hydrogen, forming a thioester. At this point, phosphate enters the picture, and phosphate is nucleophilic enough to displace the cysteine residue to form the final product. And so what's happening here is just a nucleophilic acyl substitution. One of the phosphate oxygens adds into the carbonyl carbon, nucleophilic addition, and then beta elimination of S- actually gets the original ionization state of the enzyme back, gets the free cysteine back, and generates this product. This is 1,3-bisphosphoglycerate, or 1,3-BPG, as I call it. In this molecule, we have a glycerate core, glycerate being the carboxylate version of glyceraldehyde with the carboxylate group, where we have the aldehyde in glyceraldehyde, with phosphate groups linked to carbon 1, the carboxylic acid carbon, and carbon 3 on the other end. This is 1,3-bisphosphoglycerate, and one way to think about it is it's sort of an indirect phosphorylation of carbon 1. We had a hydrogen linked to carbon 1 in glyceraldehyde 3-phosphate, but an oxidation has happened, facilitated by NAD+, to form a carboxylic acid there, and that carboxylic acid, in a sense, has been phosphorylated since the oxygen that was used in the oxidation carried around a phosphoryl group with it, right? You can think of PO43- as a phosphoryl group plus O-, which adds to the carbonyl carbon. That phosphorylation of a carboxylic acid is very important for the next step. The key idea is that 1,3-bisphosphoglycerate is an acyl phosphate, and it's electrophilic enough at phosphorus to transfer phosphate to ADP. In other words, it is exactly what we're looking for in terms of a phosphate group linked to something that's a fairly good leaving group. Good enough that the terminal phosphate of ADP can attack the phosphoryl group in this intermediate and kick off a halfway decent leaving group. And we can see that in the structure of 1,3-bisphosphoglycerate. Focus on the right-hand phosphate group and notice what it's attached to: an oxygen linked to a carbonyl carbon and carbonyl oxygen. If we were to kick off the portion highlighted in blue as a leaving group, we would end up with a resonance-stabilized carboxylate anion, a relatively good leaving group, certainly relative to an alkoxide, a much, much better leaving group. This is the structural basis of the reactivity of 1,3-bisphosphoglycerate, and more generally, any acyl phosphate is going to have this property. So I wanted to return to this table of the energetics of hydrolysis of different organophosphorus compounds. Near the top of the list are acyl phosphates, and the reason why is that they contain the phosphoryl group connected to a fairly good leaving group. Now, it's not a good leaving group by laboratory standards, but it's good enough that inside an enzyme's active site, where, for example, a general acid could protonate here, we can readily pull off the carboxylate and therefore phosphorylate a nucleophile in the process. And so essentially what's happening here is nucleophilic attack by ADP at the phosphoryl phosphorus and loss of the carboxylate leaving group. We actually see that on the product side with formation of ATP. And this is a favorable process since 1,3-bisphosphoglycerate is phosphorylating ATP. We're moving down the energy ladder here, and the overall process would have a negative delta G. So the key to phosphorylating ADP is to connect the phosphoryl group to some resonance-stabilized leaving group. And in order to generate another molecule of ATP, which is going to be required in order to get an ATP payoff, right? We've actually just generated only 2 molecules of ATP, and we put an investment of 2 in at the very beginning. In order to actually make progress and get an energetic payoff, we have to generate more ATP. And so we need to get phosphorus, or a phosphoryl group, bound to another resonance-stabilized leaving group. One way to think about doing this involves the elimination of water from this glycerate 3-phosphate intermediate that we now have. It's actually more commonly known as 3-phosphoglycerate, or 3PG. If we could eliminate water from this thing, we now have something that looks like an enol linked to the phosphate group. And so we could imagine if this fragment was eliminated, if the PO bond broke toward oxygen, this would indeed be a resonance-stabilized leaving group. And so we can think about, for example, ADP coming in and being phosphorylated by something like this. The problem with this really lies in this hydrogen, which needs to be deprotonated by a base. It's connected to a carbon that is not at all acidic. So this presents a problem. It makes this pathway mechanistically infeasible because this hydrogen that needs to be deprotonated is not very acidic. There is a way, however, to get around this. The potential alternative involves thinking about the elimination of this oxygen to form the same double bond, but with electron flow kind of in the opposite direction, if you like, from right to left rather than left to right. In order to do that, what we need to do is move the phosphate group from carbon 3 to carbon 2. This will set up a situation in which this hydrogen is now deprotonated in our hypothetical elimination process. Let me just again draw some curved arrows to highlight that. And now we're in a much better situation energetically. This is a much more feasible mechanism. The reason why is that the carbon that we're deprotonating now is now an alpha carbon. It's now alpha 2, a carbonyl group, and so it can benefit from the electron withdrawing nature of the carbonyl group, and this acidifies that position. That hydrogen at carbon 2 is much more acidic than the hydrogen in carbon 3 in our hypothetical pathway above. So that's where we're going with this, and the function of step 2.3 of glycolysis is to catalyze this isomerization of the phosphate group from carbon 3 to carbon 2. This sets up an elimination that's much easier to accomplish mechanistically in the next step, 2.4. Here's an image of the phosphoglycerate mutase enzyme, and I won't go into too many details on this, but the basic mechanism is that the substrate, 3-phosphoglycerate, first transfers its phosphoryl group to an enzymatic histidine residue to form a phosphohistidine intermediate, which is then intercepted by the 2-hydroxyl group. So rather than a direct phosphorylation by ATP and hydrolysis over here or something like this, a histidine actually choreographs it all. Histidine attacks phosphorus here and then swings around, and the phosphorus gets attacked by the hydroxyl group at carbon 2, with, of course, general acids to protonate this oxygen as the phosphoryl group is transferred to histidine, and then a general base to deprotonate this oxygen as the phosphoryl group gets transferred to this oxygen. Finally, we've arrived at this elimination of water, which is going to establish a resonance-stabilized leaving group in the substrate. And the hydrogen that is eliminated, again, is this key hydrogen connected to the alpha carbon of 2-phosphoglycerate now, 2-PG. The basic idea here is that we're going to eliminate H plus and OH minus, again, quote unquote in both cases, as acids and bases are going to be involved, to establish a new carbon-carbon double bond in the product. This is beautifully choreographed inside the active site of enolase, the enzyme that catalyzes this transformation. First of all, there's a glutamic acid residue in the enzyme's active site that assists with departure of the hydroxyl as a leaving group. And the idea here is just general acid assistance. The carboxylic acid proton associated with the glutamic acid is donated to oxygen as the CO bond breaks toward oxygen, avoiding the formation of hydroxide, avoiding negative charge on that oxygen, and forming water in the course of this elimination. The acidic alpha hydrogen is deprotonated by an enzymatic lysine residue, and this deprotonation is also assisted by the presence of a magnesium cation, Mg2+, that associates with the carboxylate, again, essentially facilitating or increasing the acidity of that alpha hydrogen. And so, on the whole, we have something that looks a lot like general acid-assisted E2 elimination, with a base removing the proton as the double bond forms, and the leaving group departing with proton transfer to the leaving group oxygen. This electron flow actually generates the product that you see up here directly. And this product contains a resonance-stabilized leaving group linked to the phosphoryl group. Furthermore, this product, if we focus just on the portion highlighted in blue, looks a lot like an enol. It looks a lot like an enol, just with a phosphate group where a hydrogen should be. It almost looks like an enol that's frozen in this form via the presence of this phosphate group. If we could somehow get rid of that, right, if a nucleophile could come in and attack that with protonation of the double bond, we would end up at a ketone. And based on what we know about enol ketone tautomerism, it seems like the ketone would be a lot more stable than this enol form. That's absolutely true. This is what drives phosphorylation of ATP in step 2.5, the next step. This enol would rather be a ketone. This metabolite, by the way, is PEP, phosphoenol pyruvate. It looks a lot like pyruvate. It just has an enol, where pyruvate contains a methyl ketone and a phosphate group linked to what's going to become the ketone or carbonyl carbon of pyruvate. Phosphoenol pyruvate is so electrophilic at phosphorus that it's not even on that table of organophosphorus hydrolysis energies that we looked at previously. It's absolutely off the charts, far more negative in hydrolysis free energy even than an acyl phosphate. In terms of electron flow, the basic idea of step 2.5, which is another phosphorylation of ADP, is the same as it was in the case of 1,3-bisphosphoglycerate. We've got a phosphoryl group linked to a resonance stabilized leaving group. Here it's an enol. Somewhere in the enzyme's active site, there's a proton, possibly associated with a general acidic residue. And in essence, what happens here is nucleophilic substitution at the phosphorus with loss of the enol portion as a leaving group. And at the same time, tautomerization occurs to generate a ketone. The resulting compound on the right is the final product, if you will, the final metabolite in glycolysis, pyruvate. We've also generated not one, but actually 2 molecules of ATP. Remember, we're running this entire step twice on the 2 3-carbon fragments generated from 1 6-carbon molecule of glucose. This means that net on the whole, right, if we look at the ATP invested and then generated, we've generated 2 molecules of ATP overall, since we've made 4 total in stage 2, and we consumed 2 in stage 1 to set up the retroaldol reaction. In addition, because NAD+ affects an oxidation and is itself reduced in stage 2, 2 molecules of NADH are also produced, 1 for each of the 3-carbon fragments generated from glucose. And that's it. We've reached the end of glycolysis. So I hope this analysis of stage 2 has shown you that the basic idea of stage 2 is to connect the phosphoryl group to a resonance stabilized leaving group. That's either a carboxylate in the first phosphorylation event or an enol or enolate in the second phosphorylation event. And it's the leaving group ability of these portions highlighted in blue, like the enolate portion here, that really drives the phosphorylation of ADP in the midst of glycolysis.